So just to show an example, if we think in dimension 1, a random variable and a double exponential distribution that decays from a center x0, the expected value is the center of symmetry, is x0. We define the same in graph. We can say, okay, we have now a probability in the space of networks that decays from some graph to any other graph on that space, and the expected graph is 0, and you can compute the variability. Okay. So let me talk a little how some of the results can be used. Outlier detection, is something important. If you have a lot of networks, thinking in brain networks, it's important to detect, okay, this is a straight guy. And how, in this space of huge dimension, how to detect it? If you have a distance from a network to the network sample, you can say, okay, if the distance is very large, it is atypical. Okay? For example, you can obtain results like this, that the distance is large for some graphs. And pay attention to these people. In the case of supervised learning, there's nothing new, because if you have a distance, the problem of supervised learning, I think everybody knows it, but let's say we have a label that can be 0, 1, or more, but let's think binary. For each subject, we have its brain network and a label that says he's healthy or not. And we train, this is the sample, and the main is to know if I have a new network, a new subject, to put the label, okay, if it's healthy or not. So if you have a distance, for example, nearest neighbor algorithm can be used. For example, something like this. Let's suppose that this two-dimensional space is the space of the networks, and each color represents some of the binary labels, and this is the training sample. Now we have one new graph on brain network, a new subject, and in the nearest neighbor algorithm, just the nearest neighbors decide if it's red or blue. And if, for example, if the three are the same color, then you predict that it will be red. And okay, and what about hypothesis testing? The ANOVA test in biology is something that is used almost all day. The ANOVA test is you want to, in dimension 1, to know if the mean value is the same in each of the three groups, for example. And it's the null hypothesis are the same, and you want to know if at least one is different. There is a very well-known test that have an statistics that compare the variance between groups within the variance within groups. Okay, so we want to develop something similar, but now our objects are networks. We have 3 groups, for example, of networks, and we want to know if they have equal central networks or they are different. And we develop an statistics that the idea is similar. We compare distance between groups and distance within groups. This is the statistics. Basically, this is the average distance from the group I to the central group of the group I, and you compare this with the average distance of all the sample to the graph, central graph E. So the idea is, if all the central networks are the same, if you measure distance between all to one central graph, will be similar to if you compare distance here to this central graph. If some central graph is different, then this will be greater than this. And then under the alternative hypothesis, this will be negative, and then you have a test for equal mean. And we show that that is consistent, and with a sample of 30 networks, we have good the asymptotic approximation is good, like in central theorem. Okay, and other good thing of having a statistics is that you can separate the problem in two. One is I want to test if the central graph are different, and if I know that if the test detects that they are different, now I can try to find which are the nodes or which are the links that are the most important, or what is a sub-network that have the largest t value. That this will be the identification problem. If I have to decide which is a subnetwork, will be the one that now minimize the t value, but over a set of a subset of nodes or links. Okay. And let me show you one application of yes. Yeah, it's computable, but yeah.
In principle, in practice.
Yeah, in practice, I will show an example with 15 nodes. but yes, it's difficult. Yeah. Okay. So this is very nice data that from the Human Connectome project. It consists of 820 subjects of 1 hour of fMRI scan, and the project also provides socio-demographical behavior and structural brain data in some categories: alertness, motor response, cognition, emotion, personality, sensory, brain anatomy. Some of these are just psychological tests, and you have the score of that. So we have brain networks, and also we have some additional data, a vector, let's say. So in principle, we have only one group of 820 networks, and what we are going to do is to divide the subject in terms of if some of the variables here are small, middle, or large. Okay? We were going to do this with many of the variables, with all, not many. Okay? So we have groups. One group is the small x, etc. And now we can try to answer this question: are x and the brain network dependent? If there is no dependence, then the central networks would be the same. And so we apply this ANOVA test.
Sorry.
Yes.
When is there a link in an fMRI network?
Okay, I will show. Okay. Yeah. So this is the typical signal of fMRI, And this is a correlation matrix, just Pearson or Spearman correlation matrix. Between each of the nodes. In this case, this is a special case because each node is not a region of the brain. Typically, you analyze, you have what is called voxel. A voxel is like a pixel in 3D, and you correlate all voxels. Here, the Human Connectome project present what is called the independent components that are regions that are coordinated. But it's the same, it's like my node is this. Okay? And once you have a correlation matrix, but once you have a correlation matrix, in order to have a network, you have to do something, and there's not much alternatives. Or you put a threshold in the correlation, or you say, okay, I'm going to compare networks of the same number of links. And say, okay, we'll compare network with 10 links, the most correlated, and that's all.